We will have 20 minutes Q&A. So can I invite the three speakers here facing the audience, and also Peter, if you're online. So we're gonna take questions. So there are several questions from online, but I would like to see anybody here in our room that have questions.
Okay, hello. So I have one question for Yue and another for Noah. I'm going to start for the one for Yue. So I was very excited when I saw your phenotype in the microcephaly organoids. I was doing my postdoc at Mootri Lab like a few years ago, and I actually used the same, I actually look at the same phenotype in other disease. So when I saw that violent plots, gave some memories. Did you see also any other kind of phenotype apart of the organoid size? Did you also look at electrophysiology, or did you look at any other stuff?
Yeah, we did look at electrophysiology. Unfortunately, there was a lab-wide incident which killed all the organoids partway through the project. So, that luckily we got the microcephaly phenotype and the rescue study done before that happened. But by that point, it didn't make sense to restart the whole thing. But we were looking for electrophysiology in the thank you, Leo, in the cells as well as the organoid. So it was on our radar.
So with that data, then it was the only kind of data you had in order to push it through the FDA and then to go to the clinic?
That's correct. That was the only efficacy data we submitted to FDA. And in fact, if you look at the N of 1 ASO guidelines, so they only apply to N of 1 ASOs, they do not require efficacy data to approve a phase 1 N of 1 ASO. So that is a very kind of special exception that FDA is making, and I think it's a good decision on their part.
Yeah, indeed. Thank you. And then also for you, Noah. Hi.
Hey, good to see you.
Yeah. I see that at the end that you are looking at 3 different strategies, no, in order to tackle the disease, no, using ASOs, using small molecules, and then using CRISPR in order to deliver to the cells. This is a little bit more speculative. What do you think is going to be the most efficient approach?
Yeah, so I think the highest probability is the existing repurposed small molecule that has the highest probability of getting into patients soonest. But the way that we're thinking about it is not that they're like separate shots on goal, but that they're a series, and we're not done. And I think each drug comes with its own challenge, right? So an ASO, it's can we get to enough of the brain regions that matter? An AAV, similar but different brain regions, potentially depending on how you did the injections. BBB small molecule, well, how okay, it's a repurposed drug, so is it just really going to be as efficacious? Do you build a custom drug? It's well, then you're going to do all the other med chem afterwards. So I think it's if you just look at what's happening with SMA, where it's multiple drugs, everybody's kind of working the problem with multiple drugs. So I have what I think will be the first, but I wouldn't say, I think it also that I believe will modify the phenotype sufficiently to get approved. But I think there'll be a series of them in the end.
Okay. Thank you very much. Other questions in the audience?
There are a few questions from online. The first question, I believe, is for Yue. It's a very practical question from Anna Rita Morella. So she said, so can you summarize the timeline and money spent during your Odyssey? I would love to have an overall idea of what to expect.
Yeah, so timeline was we got diagnosed at month 4 when Leo was 4 months old in February of 2022. It took us about 4 months or so to pull all the partnerships and all the researchers together. And from there, it was about 12 to 13 months before we got into the clinic. And we had multiple repurposing efforts going during that time as well, knowing full well that that is the fastest way to some kind of a therapeutic. Unfortunately, most of the repurposing is literature based, right? And so this protein is so little studied that there was basically one paper on TMP02. That was the paper that established it as a disease-causing gene. And so, unlike Noah, I had one paper to read, which was, it's good and bad, right? So after reading that one paper, I knew, I became the expert after, or one of the experts, right, after reading this one paper. So, but the other downside was there was very little to go on in terms of finding a repurposed drug. In terms of budget, I'll just say that it was in the millions and not the tens of millions. So it's in the high millions. And again, if you look at the Gantt chart, or there's so many dead ends and there's so many things that did not work, and that, a lot of if you take the critical path, it's in the low millions. But if you wanted to fully kind of make sure that, I wanted to make sure Leo was covered in all these eventualities, it was in the high single digit millions.
Yeah, I have a question for Noah. Yeah, so I know we did some ASO screening and identify a couple of ASOs which can increase the target protein by like 1.5, 1.7 fold. And that's your goal, right? You want to increase at that level, a small change would be enough. But since I have worked in a pharmaceutical company, in general, such a small assay window is very, very challenging for translational science. Yeah. How can we maintain such a small effect from in vitro cell culture system in the human? We see the similar effect. And also, I was wondering whether if we overshoot, let's say we overexpressed by 2-fold or 3-fold higher than expected, is there any adverse effect? So any thoughts on that?
Yeah, so a few things. So one is the ASOs that we designed. What was really interesting was while we were doing that, we found another researcher out of Loyola in Chicago who's doing the same thing. And so he had similar ASOs that were kind of in that 1.5 to 2x range. His appeared to be less toxic than ours. So we're basically in licensing that work right now. And he's right now bringing that into animals. I think it, mechanistically, so we're trying to inhibit a single microRNA. There are multiple regulatory microRNA that are targeting our gene of interest. So I just don't think you can overshoot that much. But this is like some of the other questions that our space in those 3,400 papers, for some reason, hasn't answered. Is the duplication phenotype? So there's a MECP2 duplication disease, but it's unclear whether if you introduce it postnatally that there's even a phenotype, which is like a strange, simple question that we probably should have answered a decade ago. So we don't know. The kind of general thinking is that it doesn't matter postnatally. But there's at least high confidence there's a tolerance zone of like around that 2x number, just staying below that. So I think, again, like mechanistically, it's hard to imagine how you could, just by inhibiting a single microRNA, that you're going to overshoot like 4x. It just doesn't seem possible.
Hi, thank you for the inspirational start to the day. Quick question in regards to, we've all spoke about the importance of setting precedent with the regulators and gaining access to the work and the advancements that we're making. It's a general question to all of you. It's, in your experience being so close, how have you seen that changing over time? Is it getting better as we build better tools, better data? Are we really getting to where we need to get to, which is getting these things into the patients? And I don't know how you want to answer that. It's really a broad topic.
A lot further along in the process. We are not close to IND enabling studies quite yet, but I hear from lots of fellow rare disease leaders, and it seems like there's some slow progress. I mean, everybody's doing listening sessions, engaging as early on in the process as possible. And there's still a lot of work to be done, letting regulators know what are meaningful changes to the patient community and the risks that we are willing to take because of what our kids and our families go through. So yeah, I think baby steps, but we have a long way to go.
So I think there is some progress. It's slow. It's almost like one step forward, one step back. Or I mean, 2 steps forward and 2 steps. I don't know. It's like 1.8 steps back. So it's like, yeah. So and I think maybe the most helpful thing I can say is this: physicians have a lot of leverage in these conversations. So for Leo, he had a neurodevelopmental disease, and it puts the physician in a very difficult place because when the patient has a neurodegenerative or a terminal condition, there's a much higher willingness to take risk. And I think that is probably where we will get the most progress is for the physicians, the principal investigators who are treating the terminal cases. I think we can get the most flexibility on precedent there, and then try to push that forward to other patients. In Leo's case, I was kind of, I don't know, like, I would say I didn't regret that he was neurodevelopmental instead of neurodegenerative, but I was wondering, like, if this had been neurodegenerative, would he have a better outcome because we could get the drug to him sooner, and that's just a really weird and convoluted, like, logical place to be in that the system, the regulatory system and everything has set up for the parents.
Just from my experience, we did expanded access, which is compassionate use for one of the drugs. And it was surprisingly easy. There's like a phone line you can call FDA on and get approval over the phone to start using a repurposed drug. And so I don't know that clinicians realize the power that they have, which is if they think it's in the best interest of the patient and the patient properly consents, it's a phone call to introduce a repurposed drug.
And I think that actually applies to new drugs too. If the physician is willing to put their career and their reputation on the line and say, I'm willing to go out ahead of the FDA here, they can get through these barriers.
So that's what I meant. It's phase 1. If it's been dosed in a single human, it is now eligible for a phone call to FDA.
There are more questions. So I believe the question is for Noah. So this is from Anna again. We have a community that says they really want treatment, but they don't engage as much as we would like to get the data fast. Any tips on how to ignite the community from saying they want treatment to help and do the work to actually reach them? Fundraise, data collection program, etc.
I don't think I've actually done a good job of that. I think Michelle's done a good job of that. I've been among the fortunate to have fallen into a community where a lot of people have already done that legwork and kind of community building, natural history studies. So like I got to skip that part, and I think.
Sorry, can you repeat the question?
So basically, how to encourage the community to gain data faster.
Just outreach, a lot of Facebook messages, a lot of, just community engagement. Again, engaging the scientific advisors and the researchers to communicate directly with the patient communities. That goes a long way. That really increases their engagement and motivation. But it's just, building those relationships, building trust, and taking the time to engage with the community.
I have a question for Piotr. Are you still online?
Yep, I'm still online.
Awesome. Yeah, I have one kind of scientific questions and one non-scientific question. So the first one is, I saw you have done the cell painting assay. My question, I do see you have several candidates, like 23 candidates. So how do you validate those candidates with more physiological relevant assay or any phenotype related assay? Because in general, for these unbiased assays like cell painting, you can always see some difference, but the difficulties or challenges is how to relate those principal component analysis to a particular phenotype.
Okay, so let me start by saying that in our case, what was very beneficial was that we have twin sisters. Despite they are fraternal, but they carry, the DNA from the same parents. And at some point, we decided to approach the issue from the, let's say, precision medicine point of view. So actually, we decided to only do the cell painting on Lena and Zuzia cells. And we were quite struck by the fact how fast we were able to see the phenotype using the machine learning between these two cells. Basically, at the end of the day, comparing morphology of the cells on a very different levels. And it doesn't happen very often with the rare diseases. Sometimes you look at the cells, even like the fibroblasts, and you see almost no difference or no significant difference. In our case, because probably because of the fact that we are talking about the twin sisters, yet fraternal, we were able to quite quickly distinguish the difference on the morphological level. And to your point, at the end of the day, after screening 6,000 plus FDA approved drugs, we are currently looking at the ones that are moving towards, we're looking at the drugs that are moving Lena cells as close as possible to Zuzia cells. And obviously we'll do at the end of the day some filtering regarding toxicity, regarding the side effects connected to that. And we want to filter down the list to 5, 6, which we can use on the mouse. However, if we were approach the whole issue from biased approach, meaning, hey, let's look at the publication, what we know about the PAX2 syndrome, we would never ever list these 23 compounds. They were like super surprising to basically everyone involved. We started to classify them in some mechanisms of actions. So it's not like we're not talking about 23 totally different drugs with 23 totally different mechanisms of actions. Actually, when you start to kind of gather them, you start to see some pattern. But if you would approach it from purely publication approach, so biased approach, you wouldn't pick any of them. So that's kind of the where we are with this. So at the end of the day, answering your question in a very simple way, we are going to focus on the ones that are moving Lena cells as close as possible to Zuzia cells, based on the 3 replicates we did during the final screen of the drugs from the FDA list.
Thanks. So I guess my second question is a little bit associated with the first one, and also a very general question for all of the foundation leaders. So because you guys did a tremendous job to join all these patients globally, but eventually all these studies, it's going to be case by case and focus on one patient, right? So how do you really come up with a strategy to prioritize your pipeline, and how can you scale up or make sure the resources spending in one particular patient can eventually benefit to most of the other families? I think there's I asked the question to several foundation leaders, but I know it's very, very difficult and kind of ethical related stuff. So I'd like to hear you guys as a thought leaders.
So my thinking about it was that if we could, get enough efficacy for Leo, then we could prove that these other children deserve a chance too. And then, along the way, if we publish and open source, so to speak, what we did, then that would be the, giving back to the community, and people could take what we learned and use it, themselves. I think, in terms of how it actually played out, we got the drug in Leo much later than I would have wanted. And, the efficacy is, I'm deeply grateful for any efficacy and for any, avoiding adverse events. But, it might not be so compelling to a payer or insurance. But there is the open sourcing, and we've committed to publishing our IND documents, which is often a very big barrier to getting into the clinic. And so that is the thinking that it's a learning experience, and if we give the learning to the community, then that will help every single foundation get to the next step.
So I start with that your fiduciary starts to your child, and so that's kind of how I initially approached the problem. And I made a bunch of effort to try to share what I've done from a repurposing standpoint with the community, pretty unsuccessfully. I think that the community, there are folks trying to run trials with some of these different meds that she's on. Some of them, they've already ran great trials in other countries, and it's every kid with Rett should be on some of these drugs, and it's just not the standard of care here in the States. And that's a really hard problem to change. It's like a societal thing. I think what I've seen a lot of rare disease parents is they'll spend a bunch of time trying to run a repurposed trial to prove that a drug that their kid's already on, and kind of some folks in the community are already on, works so that it can become the standard of care prescribed by doctors. Just a lot of effort to do that work. And one of the things I talked to David Fagenbaum, who's probably a lot of you guys know, but like Kurt himself of Castleman, is it's like, well, David got to the finish line, so he can help everyone get there, and like we're still running the race. And so it's a complicated dilemma, I think, to the point of prioritization for us. And so definitely could be better at sharing, but I think as we prioritize, like now I'm once you start to take on capital, you have to your fiduciaries change. If you take money from other families, then your fiduciaries change. If you take money from investors, then your fiduciaries change. And so you just have to be, I think, comfortable with what they are.
Thanks. There's another quick questions for Noah. He's asking about a compound you mentioned, a repurposed drug. Do you mind sharing?
The ones we're on?
Yeah.
There's 5 different drugs that she's on. So she's on vatiquinone, which we had to go to FDA for. That's the one that's not approved. It's a PTC drug that they're trying in Friedreich ataxia. Dojolvi, which is an ultragenics drug for medium chain triglyceride, so reduces oxidative stress. There's a doc at Emory who ran a 12-patient trial and still hasn't published on it. And at least anecdotally, it seems that it, in his study showed reduced seizures in that patient population. The third is mirtazapine. That's the one that's like blows my mind. That isn't the standard of care. There was an 80-patient study in Italy, which is pretty robust. Showed improved sleeping, reduced apneas, increased appetite, which are like amazing for a drug that you can literally, get at Rite Aid for $1. And then she's on low-dose ketamine intermittently. Again, they tried to run a study in that with Rett syndrome, but it kind of collapsed during COVID. And then the most recent one was Aricept. That was a study done by actually some of former Mootri people at Viant Bio, and they saw it as one of their top hits, kind of normalizing the organoids. They were trying to run that in a study out of Vanderbilt, and that study unfortunately just collapsed from they lost their financing from the DoD. So that's like, it's good ideas that I don't know, I didn't have to invent, they're there, and but you can see the challenges of getting them to patients over and over and over again. And I think, frankly, it requires like insane willpower to eventually get there.
We have 1 minute to take one last question. So we have online questions for Yiwei. It's also one of my curiosity. So how was AI was used in drug design and development, and particularly how I'm gonna, because in a Baby Lion project, you are trying to use the data and the experience from Leo's case to help to diagnosis and design the drug. in a faster way, right?
So I think in Leo's in making Leo's drug, Crayon Bio, using Crayon Bio's model to generate the leads was the main application of AI. There was also, I also used a few of the earlier protein folding tools to try to model the mutant protein. And I think going forward, in the last 3 years, there has just been a tremendous amount of progress in AI in biology, well, in everything, but also in biology. And so going forward, I think that the answer to that question is very different. AlphaFold 3, AlphaFold 3 came out during the course of Leo's project. It's not perfect, but it can predict interactions between proteins and non-proteins. That the performance in the model is not so good, but again, the next generation will be better, and so on. And AlphaFold 3 has competitors. They have open source competitors. There's all kinds of, Schrodinger is an older company, but there's just a plentiful number of companies who are approaching biology, chemistry, biochemistry, even cell biology. There's an effort at CZI at building an in silico cell model, and that will just change, it will change everything. And so I'm really excited for, where AI is now. Like, there's also the Baker Lab, they can predict protein binding from the sequence alone, the sequence of amino acids alone. And they can get up to 50%, they can get 50% success, at just picomolar, dense. Anyway, and it's incredible, right? So that's where we are already. And so, even a year or two from now, I look forward to the progress.
Thank you. So let's closing up this first section, and